Hey everybody, in this video we are going to learn about how the Agrobacterium system for transforming plants can be used to mutate specific genes in a plant. So we talked about Agrobacterium already and learned how we can insert a tDNA from the TI plasmid in Agrobacterium into a plant, sort of randomly in the plant genome. Now we want to, what if we don't want to insert a gene into the plant? What if we just want to mutate a gene? So not put a new gene in a plant to give it a new trait by expressing a new gene, just keep a plant from expressing a gene that it normally has. So one example would, a classical example is from a plant called Brassica napus. Now Brassica napus is a plant that is well known for making rapeseed. And rapeseed is high in oil, and in the middle of the 20th century, early 20th century, the oil from rapeseed was used for, I believe it was used in like motors and engines and things like that. But you couldn't really cook with it because the oil was high in a compound called erucic acid. And apparently erucic acid is toxic at high levels, or the levels that were in rapeseed. So, it wasn't edible. So through breeding programs, plant breeding programs in the 60s and 70s, 1960s, 1970s, a low erucic acid variety of rapeseed was created. And so how do they do this? Well, they probably do this by, random mutagenesis, like looking for random mutations, using UV light or other chemical mutagen, and then selecting for plants that just, randomly, from that mutagenesis process make low erucic acid. And this is what we know as canola oil. So this is where canola oil comes from. And just a quick Wikipedia search suggests that canola stands for Canada, for can, O for oil, and then LA for low acid, so low erucic acid. So plant breeding programs are extremely labor intensive because you're screening thousands and thousands of plants, hoping to find, one mutant that, at least one mutant that shows the phenotype you're interested in. And you have to hope that low erucic acid doesn't come along with other phenotypes that would be bad for, say, producing high amounts of oil, right? Like maybe low erucic acid often comes with not making many seeds where the oil is found, and that would be bad. That wouldn't be very profitable. So wouldn't it be nice if we had a targeted approach where we could mutate a gene that's required to make erucic acid and not mutate anything else in the plant? Well, we can do that. We can do that with Agrobacterium, but we need to adjust the Agrobacterium system to mutate a specific gene. And we can do that by combining the Agrobacterium system with CRISPR-Cas9 technology. So let's see how this works. Now, let's review the Agrobacterium TI plasmid. And so we are going to use a recombinant TI plasmid. So a recombinant DNA, again, is just a DNA molecule consisting of DNA from multiple sources. And in this TI plasmid, we'll have our virulence genes over here, and we'll have an antibiotic resistance gene over here. And here we'll have an origin of replication. Now remember, the antibiotic resistance gene is not normally found in the TI plasmid, but we put it in there so we can get this plasmid back into Agrobacterium after we take it out and put other genes in it. So, and what will we want here in the tDNA region between the left border and the right border? So remember, anything in here is what's going to go into the plant. So we probably would want some sort of gene for herbicide resistance. And then, because we want this to, we want to put a tDNA into the plant that has genes that can mutate a specific gene of our choosing. So here, let's put the gene for Cas9. So Cas9 is a nuclease, and it's the nuclease of the type 2 CRISPR system that we talked about in a previous video. And so this gene is going to express Cas9 in the plant, in plant cells. And then over here, we're going to have another gene that expresses a guide RNA. Guide RNA gene. Okay, so what this gene does, it expresses a guide RNA that the Cas9 can pick up and use to mutate a specific gene. And what gene will it mutate? Well, it will mutate the gene that is complementary to the spacer sequence of the guide RNA. So those first 20 nucleotides of the guide RNA molecule that we talked about in a previous video. So in our example, what we would do, we would design the guide RNA that we put here to target that erucic acid gene and mutate it. So the Cas9 will mutate that gene. Okay, so if this is our recombinant TI plasmid, what we would do is we'd have to put this into Agrobacterium, and then we need to use Agrobacterium to insert the tDNA into a plant. Maybe it'd be canola or another plant. Not canola, but Brassica napus, or another plant that we're interested in using for oil production. And how do we do this? So see a previous video. I think the previous video we talked about this. You could dip the flowers of the plant into a solution containing the Agrobacterium. Agrobacterium would then mix with the reproductive structures. The tDNA would get into the seeds. You'd plant the seeds, and then you would treat the seeds with herbicide, and only those that are resistant to the herbicide will continue to grow, and those that continue to grow would have the tDNA, because the tDNA has the herbicide resistance gene. So select plants. Now we can call these transgenic plants because they have the transgene, which is the tDNA. So based on herbicide resistance, okay, so where did that tDNA insert? Now Agrobacterium, when it inserts a tDNA into the plant genome, it inserts it randomly. Inserts it into random locations. Okay, so let's imagine what that looks like now in that plant. So let's take a random chromosome in that plant, and let's say the tDNA inserted here. Now I'm gonna designate that allele that has the tDNA with a capital T. So capital T equals tDNA. Now let's say this is a diploid plant. The allelic location on the homologous chromosome Remember, if this is a diploid plant, the tDNA inserts on one chromosome, that other homologous chromosome will not have a copy of the tDNA. We're going to call that allele little t. Now, let's say that gene we are targeting because that gene, we think it controls erucic acid production. We want to eliminate erucic acid from the plant. That's who knows what erucic acid does. Maybe it deters insects from eating the seeds. And so that's why the plant makes it. It's a possibility. So we want to get rid of that. Then you'd have to worry about, well, if you grow this plant, are insects just going to eat the seeds? We'll worry about that later. So, okay, so let's say the erucic acid gene is down here on this other chromosome. So diploid organism, so we have 2 of each chromosome. Now we're going to call this gene E for erucic acid. I'll use lowercase e with a plus. I'll put a plus up here next to this little t right here to show that this is the wild type chromosome. The big T is the chromosome with the tDNA. E plus, this is the wild type erucic acid gene. Okay, so the tDNA is here. It has herbicide resistance. It has the gene for herbicide resistance, but what else does it have? It also has Cas9, the gene for Cas9, and the gene for the guide RNA. So as soon as this tDNA gets in here, Cas9 is going to be expressed, and the guide RNA is going to be expressed. Cas9 is going to pick up that guide RNA. It's going to search every chromosome, every molecule of DNA. Eventually, it's going to find E+. In the guide, remember that guide RNA was designed to find E+ and mutate it. So E+ is now going to become E-. Well, let's go En for E null. We're going to hope it mutates it in a way. We talked about this in the CRISPR, in one of the CRISPR videos. Cas9 cuts the DNA, and then the DNA is typically fixed by non-homologous end joining, which often puts mutations into the gene that disrupt its function. So we're going to assume that's what happened here. Mutated the gene, creating null alleles. So this gene is no longer functional. It's going to get both copies too, right? There's no reason to suspect that it would get one but not the other. So Cas9 is going to keep targeting these until they're mutated. Okay, great. So we have our result. We have our plant that no longer has the erucic acid gene and hopefully doesn't make erucic acid if our predictions were correct and this gene is in charge or is required to make erucic acid. That plant will no longer make erucic acid. But we do have one problem now. We have this tDNA here still that has Cas9 and has the guide RNA for this gene and has the gene for herbicide resistance. And there are still major regulatory hurdles, and perhaps there should be major regulatory hurdles for using this type of plant right here that has a tDNA, that has Cas9, a guide RNA, and herbicide resistance. It's not easy to get approval to put this and plant this in a field. So what we have to do is we have to remove this. We have to remove this tDNA. And as long as we remove this tDNA, well, we can plant this in the field. And why is that? Well, because these mutations right here, these theoretically could be constructed by just random mutation. You know, you'd have to work for, maybe 10, 20 years and spend hundreds of millions of dollars to do it. But eventually, you could get this using traditional breeding techniques. Just random mutation and screening plants until you find a random mutant that happened to have mutations in a gene that controls erucic acid production. So if we can separate these mutations from the tDNA, well, this plant won't count as a genetically modified organism. And we could get approval to plant this in a field. But how would we separate this? How could we do that? Well, one thing we could do, well, maybe this plant is self-fertile, right? So we talked about pea plants, how you could cross the plants to themselves. Maybe this plant is similar. So we could just self-cross it, and then what would that look like? So let's see. The genotype looks something like this. Now we're going back to the beginning of the course, classical genetics, right? So here's the genotype, homozygous for the rusic acid gene, homozygous null, and heterozygous for the tDNA containing gene or chromosome that has the tDNA. We have the tDNA chromosome and the wild type chromosome, T+. So we cross those together, what happens? Now remember, it's been a long time since we've done these, but we can separate them and take them one character at a time. And so homozygous null, the homozygous null, this means all the offspring are going to be homozygous null. And then we have heterozygous cross to heterozygous for the tDNA. So that means 1/4 are going to, we could do the Punnett square if we wanted to, but maybe you remember this. 1/4 are going to be homozygous for the tDNA. 2/4 are going to be heterozygous for the tDNA in the wild type allele of the wild type chromosome. And 1/4 are going to be homozygous for the wild type chromosome. So combine these with the forked line method. We can just put a 1/1 for these. And we have our 1/4 to 2/4 to 1/4 ratio. TT, big T, little t plus, and little t plus, en. Okay, so this is our plant. No longer makes erucic acid, no longer has the tDNA, and okay, this is our plant. This is the thing we're going to want to grow in the field, and this is yeah, okay, so this is suitable. It doesn't count, according to current U.S. Department of Agriculture and FDA rules, does not count as a genetically modified organism. Even though we used genetic engineering to get to this point, it doesn't count as a GMO. And I believe the reasoning is it would be possible to create an allele like this using traditional plant breeding techniques. It would just take dozens of years and hundreds of millions of dollars, unless of course you got really lucky. So okay, so this is a way we can combine Agrobacterium with CRISPR-Cas technology to obtain specific mutations in specific genes while creating a plant that doesn't count as a genetically modified organism. So that's it for that video. I think in the next one we are going to take a look at CRISPR-Cas9 technology in applying that to mammals. Okay, see you in the next video.